Well, good morning or afternoon, whatever time you're watching this thing, for this Nestle Nutrition Institute program titled The Role of Nutrition Care in Mitochondrial Health. As many of you are aware, the mitochondrial health has become a big hot topic. In the last couple of years, we've seen hundreds of articles, hundreds of concepts of what the mitochondria really is doing, and can we change nutrition is our focus today. For our sponsor disclosure today, we have financial support for this presentation was provided by the Nestle Health Science. The views expressed here by myself and the other presenters are those of ours, and do not necessarily represent Nestle's views. The material herein is accurate as of the date it was presented and is educational purposes only, is not intended as a substitute for medical advice. Reproduction or distribution of these materials is prohibited. So this is our crew today. As myself, I'm Robert Martindale. I work at Oregon Health Sciences in Portland, Oregon, where I'm a professor of surgery and basically an active clinician. The other two guys are real scientists. Brett Goodpasture comes to us from Adventist Health. He's the senior director of the Scientific Institute of Advent Health. And he's worked with the pathophysiology of human obesity, insulin resistance, and diabetes, and certainly actively involved in how the mitochondria are involved in this. He's got an interesting background where he did his BS in biology at Purdue, and then went on and did a pre-doctoral fellowship in Maastricht University in Netherlands, which, as many of you know, is the place to go and study metabolism in Europe, and really a world's leader in the concept of metabolism. Then received a PhD in bioenergetics from Ball State University, and now has been very active in the work of mitochondria, exercise physiology, obesity, etc. Dr. Cecchini is from Mayo Clinic. He's did his training in Brazil, and now is currently professor of pharmacology and assistant professor of medicine and professor of anesthesiology. And there, Dr. Cecchini certainly has been active in the area of mitochondrial physiology, and certainly is now the co-director of the Mayo Clinic Mitochondrial Center. So his whole life revolves around mitochondria, and I think you'll see that in this presentation. It's a very exciting area. So we'll move on from there, and the program objective today, as you can see here, really is to go over some mitochondrial physiology, because for many of us, we thought of the mitochondria as just to make ATP. And we'll see we'll delve into some of the other functions the mitochondria have. Well, my talk today is probably the simplest of the three. Certainly, I'm not a mitochondrial expert. I like to fool around with it. I like biochemistry, so it fits for me. But certainly the other two guys are the real experts. My disclosures for this program are really none. I got other related disclosures, Nestle Nutrition Fellowship. I'm one of the faculty for that, as well as a couple of hernia companies, which I've done some work in the past with. So you can see here my objective today is really to go over the function of mitochondria. So let's get right to the science of this. I mean, I think most of us we all talk about mitochondria, but I have to tell you, we know little about it. Even with my PhD in biochemistry, we really are still trying to understand the mitochondria. We know they're both in plants and animals, and various cells in your body have different amounts of them. The red cell, for example, has only no mitochondria and has to use glycolysis for its energy supply, whereas hepatocytes and cardiac cells have a large number of those. Evolutionarily, many people believe, and I think the current concept is, that mitochondria arise from a symbiotic relationship from probably a bacteria which got intracellular and then became really the donor of energy for the cell, and it became a very nice symbiotic relationship which fits. We know this really from now being able to genetically test for mitochondrial DNA, which is different than the rest of the cell DNA. The mitochondria has an interesting life cycle. It can fuse, it can undergo fission, which increases the number of mitochondria, fusion, which increases the mitochondrial mass. It can then, under certain cellular signals, it can undergo mitophagy, which is degradation, sort of like the cellular autophagy. It undergoes individually by mitophagy. And so it's a very exciting and dynamic life cycle, which we see in the mitochondria. We see mitochondrial dysfunction in many of our clinical diseases, which we deal with every day. We know that ICU patients have about a 50% reduction in ATP compared to healthy control. We know that excess free radical oxygen negatively affects the electron transport chain, so our ability to make ATP, that high energy phosphate bond, is altered. We also know that a leak in the mitochondrial membrane integrity sets off a leak that allows mitochondrial DNA to leak out, which serves as one of the danger associated molecular patterns. Basically tell us that our body is breaking down and we need to protect it if at all possible, a danger signal, you might say, for the body. And so the classic understanding, on which if I ask even my medical students when they come on my service, we talk about the ATP, and they all say right away, it makes ATP. That's what it does. It makes energy for the cell. That's absolutely right. But in the last 15 years, we've really realized that our current understanding needs to really grow. And we need to understand that the mitochondria are critical in cell signaling, gene regulation, cell growth, absolute calcium metabolism, influxes of calcium in and out of the cell, modulating cell death pathways and autophagy, and we already talked about mitophagy, as well as multiple other functions. So again, a very dynamic chain. If we look at the mitochondria, I'm not going to bore you with too many details, but I would like to just a couple of things I want to talk about here is we have, remember, a membrane really inside of a membrane. We have the outer mitochondrial membrane, which is freely permeable to small molecules like ions, amino acids, and sugars. They can get into this out through the outer mitochondrial membrane, but they don't get into the inner mitochondrial membrane without specific transporters. The inner mitochondrial membrane is highly impermeable. So we have a moderately permeable and an inner mitochondrial membrane non-permeable, and that allows for the electrochemical gradient, which then allows us to make the ATP in the respiratory chain. Mitochondrial DNA sits in the inner mitochondrial membrane, and the Krebs cycle, the TCA cycle, takes place at the inner mitochondrial membrane. So you can see we have to transport carbons back and forth across that membrane to be able to generate the electron transport system and ATP. This nice picture of a mitochondria up here. To make that mitochondria work, what do we need for optimal function? The electron transport proteins. We've got to have those proteins for every phase of the multiple steps of the electron train. We've got to have the Krebs cycle functioning, and that way allows for stable membranes. Without stable membranes, the mitochondria falls apart, and the cell's ability to make proteins or make energy drops. So then in critical illness, let's take that for an example. What do we see in critical illness? We see a depletion of the required electron transport chain protein. So maybe we should give more protein. That's been a thought. Maybe not. Leakage of proteins across the normal impermeable inner membrane. Remember, that's impermeable. That's critical to remember because we need the electrochemical gradient across that membrane to generate the energy. Incomplete delivery of electrons and molecular oxygen. That generates superoxide, hydroxy radicals, etc. So this electron transport chain for oxidative phosphorylation is key. It requires multiple factors there to make it work. As you can see here in these mitochondria packed into cells, if you look at a normal non-septic control here, you can see here, and then here we see a septic survivor. And look at the mitochondria differences. They've got these big gaps, as you can see. And this here, we see it by this nice work by Owen recently, showing a normal mitochondria, an aberrant state mitochondria, and then certainly maybe even a dying mitochondria here as an empty state. This is where we get into what's fusion, fission, mitophagy. Different cells get signals depending on the state they're in. Now, there's been some very nice work done by Zuden Putticherry trying to understand this mechanism. And why is it we can't use more energy? We know that there's decreased mitochondrial biogenesis in the critical illness, as I mentioned already, and there's dysregulated lipid oxidation. And remember, lipid oxidation is the big key because that's a huge energy producer for us. So at the end result is we have reduced ATP generation, skeletal muscle wasting, as we see in the ICU setting, with a significant inflammation of that muscle, and we see intramuscular inflammation impairs anabolic recovery and alters lipid utilization in the mitochondria. So now what do we have? In a septic state then, in whatever we look at, early critical illness, the mitochondria may in fact be the source of the problem. So what do we see? We see decreased ATP and increased lactate, which we all use clinically to measure, and gives us all these issues: insulin resistance, loss of muscle mass, energy deficits, protein deficits, GI dysfunction, oxidative stress. And the big one, which is hot today to talk about, is inflammation and increased catabolic response. So what do we do with that? Well, okay, if we can just get more energy to the cell, that's the answer. So is more macronutrients the answer? Is more micronutrients? Clearly in nutrition, that's what we want to do, right? We had a big push years ago to get the goal as fast as we can. Maybe that's the answer for the mitochondria. Well, what happens when you do that? Remember, this slide was loaned to us from Zuden Putticher. Very nice slide looking at glucose to pyruvate. Glycolysis takes place in the cell cytoplasm. Then here's the catch. In the inner mitochondrial membrane is where we get the TCA cycle or the Krebs cycle. Remember, the whole glucose going to CO2 and water gives us 38 ATP total, okay? Glycolysis leads us only to ATP. You can see a big discrepancy there. So the key is, okay, let's drive the Krebs cycle. The logical approach then is give it more energy, carbohydrates, proteins, lipids, to drive it. Well, unfortunately, it doesn't work. We've learned that the hard way. When TPN first came out, we said, aha, we finally have the answer. We can give TPN and give calories on day one of critical illness, and we learned that was detrimental to the patient. And then we learned that even enteral feeding full calories first day is probably detrimental because we cannot get those two carbon fragments into the Krebs cycle. So we have decreased mitochondrial beta oxidation. We have pyruvate actively going on, glucose to pyruvate, but the pyruvate can't get into the Krebs cycle, so it goes to lactate. Lactate then diffuses out of the cell and becomes an energy problem and acidosis problem. Okay, well, if macronutrients, carbohydrates, fats, and proteins don't work, what about maybe we should give more micronutrients? Because these micronutrients are certainly cofactors in many of the steps in normal metabolism. So we've read a lot recently about B1, Now there's associations with B1 and increased mortality in the ICU, and B1 cardiac surgery makes no difference with no change in lactate. So the data is all over the place, but the bottom line, huge doses of B1 do not seem to be the answer. What about riboflavin? Again, we clearly see altered plasma FAD and riboflavin levels in ICU population, but remember, we don't know what a lower level means. Maybe that's the body's way of protecting itself. B12, same thing. We've seen both adverse events and decreased by adverse and beneficial benefits with the B12. What about vitamin C? Well, there's one. Everybody needs vitamin C, we think, because it's an antioxidant. It's going to prevent those oxygen radicals from injuring the cell. Well, in reality, when we actually look at Olympic athletes, we know that when we give them certain levels of vitamin C with their training, when we give them too much, what do we see? Decreased mitochondrial biogenesis. So clearly, to make new mitochondria, the mitochondria need to be stressed a little bit, so they realize they need to have more energy, so they make new mitochondria. And we can shut that off by giving too much vitamin C. And we know that in these athletes, that was seen. Their performance drops off when we give too much vitamin C. We see lower maximum oxygen consumption. Vitamin D and ICU trials, everybody thinks a hot topic, vitamin D now, it showed no big benefit in ICU trials. Vitamin E, maybe if we can make that soluble to get to the inner mitochondrial membrane, there's some new drugs associated with vitamin E that may work. We don't have enough data yet. Oh, I see. Maybe it's a trace minerals then. Well, what about selenium? Selenium has gone from just discovering as an essential trace element in the 70s and 80s to being the cure for all of our problems. Because remember that selenium is a cofactor for superoxide dismutase, for glutathione peroxidase, all the antioxidant enzymes. We know stressed ICU patients have more oxidation problems and more, free radical oxygen, but has it really shown to help? In fact, not very much benefit. Randomized clinical trials and systematic reviews shows supplementing selenium and sepsis shows no benefit. Unfortunate. Zinc, similar sort of thing. Some associations with lower level, but does it really change survival? Probably not. We now go into things like carnitine, lipoic acid, melatonin. Now we're looking maybe more at some of the more scientific approaches here that maybe it takes a little bit more. Melatonin in animal models has got some beautiful data showing benefit in septic model, rescuing the mitochondria from oxidative stress, increasing the electron transport system and ATP production. Again, animal models, but we know also melatonin is low in critical illness and low levels are associated with more sepsis. But remember, we got to be careful. Just having low levels or replacing doesn't mean it's going to work. Remember that mitochondrial membrane transport across it is very critical. Carnitine's another one we've known for years. Carnitine's required to get long-chain fatty acids from the cytosol into the inner mitochondrial membrane so they can push oxidative phosphorylation. So does it work? Some people say it does, but most reports say not consistent data. So again, lots of interesting topics, but the question is, why hasn't it worked? So now we've shown macronutrients probably don't work, giving large volumes. We can't push glycolysis anymore. We can't push, some things. What about giving a mixture? Well, maybe that's the answer, but we don't know what that mixture is yet. Well, how about adding lipophilic side chain to make it more soluble for that inner mitochondrial membrane, which is pretty impermeable, to let it get in there? That looks like it has promising, but needs work. Metabolic reprogramming may be the answer. Getting the cell to just start using different energy sources. What about timing? Maybe we should not give so much early. We know from the parental and the enteral experience and all of our recent ICU studies, partial calories seem to be equivalent to full calories. And we've got the Rice study in 2012 was the first, but now we've got a Robbie trial, the EAT ICU trial, the ANSEX trial, the Target trial. All those trials showed that we don't need to give full calories in the first day or two. We can wait. More calories is probably worse. So that didn't work. What about cycles, feeding and fasting? Some people would argue the whole ketogenic idea that maybe with ketosis going up, we can push that oxidative phosphorylation through the ketone bodies. Correlating feeding with protein with resistance exercise. This is certainly one of the areas, and Dr. Goodpasture will show us that. He's truly one of the world's experts in this area. The microbiome. What about this staged approach? Slow ramp up and use biomarkers when to start giving more nutrients. That's probably the answer, but we don't know what the biomarkers are yet. Individualized and personalized nutrition prescription. Probably one of the answers there too. And this idea of metabolic reprogramming is hot. This is the idea where we use, this is some animal models here, where we actually can show that if we block glucose production, we improve mortality. So maybe that enhanced glycolysis is detrimental. And again, we've got lots of data there looking at individual animal models with kidney injury, liver injury, cardiac injury, showing that possibly pushing glucose may not be the answer. And do we have any human data there? Some animal data, this is very nice, interesting data. And when we look at maybe we should individualize these things. For example, what do we do with fever caused by basically a virus, a bacteria versus a virus? Well, look at this. In this model, a mouse model, they compared viral and bacterial infection in feeding and fasting. Okay, we've always heard the story, starve a fever, feed a cold. Okay, well, in this case, they did two models of bacterial infections. listeria, and bacteria commonly used in the lab for inducing sepsis, or lipopolysaccharide. Two different models, two different groups of animals. They showed that anorexia, or not eating while giving nutritional supplements, gave poor outcome. So not eating was more benefit. Anorexia, animals not eating, had a better outcome than those they force fed. So glucose was needed to show the detrimental effects. So ketogenesis was beneficial. So interesting that when we have bacterial infections, maybe we're doing the wrong thing by pushing calories. But what about viral infections? Here, they use an influenza virus or a polyol-C, which is an artificial viral model to get viral sepsis. They showed nutritional supplemented protected against mortality. And when we block glucose in this model, it was lethal. So we have to think now, maybe we've done it wrong for years. Maybe we've given everybody in the same ICU the same nutrition prescription. Maybe we should now individualize those and metabolically search for the answer and say, this is a viral infection. We need to give this person glucose. This is a bacterial infection. Maybe we need to hold off on nutrition for a while, give it 24 to 48 hours, and then we can start to give it. So we have to rethink our thought. We have to rethink what we're doing now as we're learning more about the mitochondria. So this is a proposed concept that came from a nice paper in Nature from Dr. Harris. As you can see here, I put my little symbols in there to make it fit. So in the first couple of days of critical illness, we can make sure we're not giving excess nutrients. That appears to be a trend, okay? We need to give our basic things minimal nutrient support, but we need to think about the microbiome. We need to think about micronutrients, but again, not excessive amounts. And then when we're in a hyperinflammatory phase, 48 to 72 hours maybe, we don't know the exact timing, then we start considering extra antioxidants to decrease the damage from reactive oxygen species. We need to process the increase of protein to support the acute phase response to illness. And then we're in the recovery phase. Here's where we need to do exercise, increase protein, resistance exercise, I mentioned, maintain vitamin and trace minerals. And here we may want to start considering things like EPA and DHA, because we know the fish oils make resolvins or specialized pro-resolving mediators, which actually enhance resolution of inflammation. Remember, inflammation is an overriding theme here in all of this. So I think the problem here is if we're going to use this staged approach, we should have biomarkers, which were not figured out yet. So I think that's coming. But this approach at least gives us some concepts to build on. You know, the future is very exciting, adding lots of interesting lipophilic side chains looks good. The sirtuins looks very interesting. Animal models, we've shown protective of cardiovascular, metabolic diseases. You know, the PPR gamma coactivator group of proteins, again, another exciting areas are major regulators of mitochondrial DNA-dependent enzymes and proteins. Early work both in animals and humans show promise here. There's some human recombinant transcription factors for mitochondrial protein. They may reduce mortality in animal models, but we haven't seen human stuff yet. And even thoughts like in mitochondrial transplantation. This is mostly done in vitro cellular models, but you can they are taken up by the myocardium and actually can increase myocardial function, contractility. So there are lots of exciting areas. The future for us in nutrition is going to be focus our thoughts on nutrition, on the mitochondria. So now, what about nutritional modulation of mitochondrial function? Where should we be? Remember, most current data is extrapolated from human biopsy data in vitro and animal studies. That's where we're learning the science. Okay, now when we start talking about this specific illness, we need to be very careful, okay, that we have lots of agents, but we're very naive if we think we can just give more carbohydrate or more vitamin B1 and we're going to make a difference. It's a very complex metabolism, which is going to take a lot of computational biology to help sort out. There's lots of knowledge gaps. What's the ideal level of each component? Do we need a cocktail of these components? What are the serum levels? Are they not useful? What about intracellular? What about intramitochondrial levels? Maybe that's where we need to know. You know, altered protein binding or distribution. So again, we've got a lot to learn before we can talking about specific nutrition. For now, I think we can say deliver moderate amounts of macronutrients with a relatively slow escalation over the first few days of illness. Judicious micronutrient delivery to promote antioxidant defenses. Early enteral nutrition support the microbiome, but again, at a relatively modest level. We don't need to push it to day one. Meticulous glucose control, this is key, preventing the end products of glycolysis, which become toxic to the mitochondria. Additional protein in acute and recovery phases are key, giving more protein as the patient is doing better. Again, resistance exercise goes along with this. And again, we will see those results coming out now. We're starting to see those. What's the optimal function? I think we don't really know, but we're starting to get some techniques to be able to use that. Continuous indirect calorimetry, looking at in vivo substrate oxidation, near infrared spectroscopy, and that gives us in vivo muscle oxygen consumption. So those are real-time measurements. Phosphorus NMR spectroscopy will measure total high-energy phosphate bonds within the cell in real time. This is where the action is. What's happening minute to minute, second to second in the cell as we're seeing in different illnesses and different recovery phase or aging and all these things. And of course, proteomics, transcriptomics are helping along this case. Now, our next slide, our next presenter is Dr. Goodpasture, as I mentioned, scientific director at Advent Health Research Institute, certainly a world leader in mitochondrial function. His expertise in obesity and insulin resistance and exercise-induced physiology and mitochondrial function are fabulous, and I think you'll enjoy his talk. Thank you very much.
Hello, everybody. It's good to be with you today. I'm going to talk to you about innovative nutrients supporting cellular health during aging. First of all, just to disclose, I'm on an advisory board for both Nestle and Emeon Incorporated. So to set the stage for today's talk, I want to go through some brief objectives with you. First of all, I'm going to discuss the role of mitochondrial energetics in human aging, and this will be a common theme throughout the talk. I also want to discuss or interrogate the roles of both obesity and exercise in aging. And you'll notice that exercise or aging is in quotes here because we often think of aging as this unmalleable or unchangeable factor. But as you'll see in the talk, that we think that we can actually mitigate or sometimes even reverse or prevent some of these so-called aging effects. And then thirdly, I want to highlight some of the data supporting the roles of both calorie restriction for weight loss and exercise in mitochondrial biology of aging, really highlighting the confounding variables of physical activity and obesity or body fatness in mitochondrial biology. And then lastly, discuss some potentially important nutritional strategies to enhance mitochondrial energetics in human aging. So it's well accepted that aging is associated with the decline in physical function and an increase of risk of certain diseases like diabetes, like cardiovascular disease, Alzheimer's disease, dementia, etc. The declines in energy, strength, and resilience are also commonly reported related to physical dysfunction as people get older or age. First of all, fatigue is a commonly reported symptom or condition of aging. Approximately a third of adults over 51 years of age report some form of fatigue. Also, muscle strength declines by about 1.5% per year between 50 and 60. And then after about age 70, there's a rapid decline in muscle strength, about a 3% decline in muscle strength per year after the age of 70. Also importantly, age is associated with increased oxidative stress and a reduced immune response. So approximately 80% of adults over age of 55 have at least one of these chronic conditions. As you're going to see during the talk, that mitochondrial biology or function is integral in many of these both self-reported and measured indices of aging. For example, we've reported in our own data that when you look at mitochondria function on the y-axis here, older adults who report low fatigability have higher mitochondria function compared to those older adults who report a higher fatigability or have higher fatigue index scores. And these data really implicate the role of skeletal muscle mitochondrial energetics or function in measured and self-reported fatigue in older adults. Now, this mitochondria function is implicated in what we call age-associated cellular decline, or AACD, which in a nutshell really describes the cellular changes underlying the aging process and development of age-related conditions. In other words, these aging processes and age-related conditions in people have to start with a cellular basis for this dysfunction. And importantly, these cellular changes often precede the clinical signs. And this is an important component of what I'm going to talk to you about today, because it'll be important to identify pathways and targets for the aging process so we can really head these off before they get to the point at which the person actually feels or is symptomatic for the condition. So the key manifestations of this age-associated cellular decline to target for intervention certainly would be the decline in self-perceived energy, again related to the fatigability, engagement in physical and social activity, and importantly, declines in mobility, how well people get around and get along, relating to their muscle strength, muscle function. So we really believe that intervention should target the fundamental mechanisms of aging, including mitochondrial dysfunction. Because at the root of impaired function of the whole person likely comes down to the cellular components of decreased function with aging, and even more so the energy metabolism aspects of the cell, which would include mitochondria dysfunction. So mitochondria dysfunction could precede the cellular decline, which is certainly related to impairments of the organ or the system that ultimately relate to decreased function of the whole person. Now, there are many factors that contribute to this age-associated cellular decline and mitochondria dysfunction. Here are just a few that we're going to talk about today. Poor nutrition, for example, sedentary lifestyle. Some of the data that I'll present today will highlight the effects of exercise, weight loss for improved metabolism, and then also talk to you a little bit about some of the nutritional interventions, more novel, more recent nutritional interventions that may ultimately be linked to these mechanisms of mitochondria dysfunction and age-associated cellular decline. For example, mitophagy activators, we're going to talk about some of these nutritional compounds that may impact mitochondria biology here. NAD precursors, some of you may have heard about NAD boosting. We're going to talk a little bit about that, although the speaker after me, Dr. Cheney, will talk more about NAD biology and NAD boosting. And then lastly, talk about mechanisms of oxidative stress in aging and how nutritional interventions may be able to mitigate or potentially even prevent excessive oxidative stress in aging. One of the tools that we've used in our research over the last decades has been to acquire skeletal muscle tissue in human volunteers through a needle biopsy method in which we can obtain anywhere between 100 and 200 milligrams of tissue. It's a needle that goes through the skin and the fascia that obtains the muscle. And as you can see from this slide, we can get a lot of good information in a fairly small amount of muscle tissue from a volunteer to learn a lot about muscle biology. So some of the data, including this, will highlight this. So we've measured mitochondria function here on the y-axis in these muscle biopsies. As then it turns out, the mitochondria function in the skeletal muscle is really not strongly associated with age per se. You can see that when we look at subjects from age 20 all the way out to age 90, there's a significant association, but you can see that it's a very weak association. What we typically see is that there's a stronger association between BMI, or the level of body fat or obesity, with mitochondria function with in these subjects. You can see that if you're overweight or obese, you tend to have a lower mitochondria function than if you're normal weight or lean. But the strongest association with mitochondria function, as you might imagine, is with physical fitness. So we can actually measure physical fitness. We essentially bring people in to the exercise testing lab. We can put them on a stationary cycle ergometer or a treadmill. We have them exercise to their maximal capacity, and we measure the oxygen uptake. And this is the gold standard of their physical fitness, their VO2 max. And the mitochondria function in the muscle is fairly strongly associated with the maximal exercise capacity for the VO2 max. Much more strong than the association with age. So the take-home message from these data are that age per se is really not a strong correlate of mitochondria function in muscle, but it really comes down to chronic exercise and physical fitness that's the primary driver. What you see here in front of you are 2 electron micrographs taken from 2 of our volunteers' muscle biopsies. You can see on the left, this is a master's athlete, so someone in their 60s and 70s who's maintained a high level of activity. You can see lots of mitochondria, these dark organelles here, compared to the person on the right who's the same age but doesn't exercise. Very few mitochondria, and what mitochondria they have are very small. And I can tell you, and I don't have the picture here, but if you put up a young athlete next to this old athlete, it looks essentially identical. And this is important data showing that from the muscle perspective, that if you maintain a high amount of physical activity, the muscle can be maintained to a very healthy state with regard to the mitochondria function. And we can actually quantify this, and it's interesting or sometimes useful in this case to actually see the numbers. You can see the mitochondria content in the muscle cell, that's about 5% of the total volume, and the athletes, which is double the amount you see in the sedentary subjects who don't exercise. So chronic exercise is a powerful indicator of how much mitochondria you have in the muscle cell. And so if you're asking, can I do anything about my mitochondria now that I'm older and sedentary and I want to start an exercise program? Well, the answer to that is emphatically yes. So we just completed a randomized controlled trial of health education control, weight loss only, and then the same weight loss program with an exercise program in older, obese, insulin resistant, and sometimes obese patients with type 2 diabetes. The take-home message there, I won't go into all the details. We look at the various components of mitochondria function. The take-home message is really consistently the same, and that is the weight loss by itself in the middle bars here doesn't really do anything to mitochondria function until you add an exercise program. It's only with the exercise program that you get this robust improvement in mitochondria function that does not occur with calorie restriction to induce weight loss by itself. And just to say that these data were led by a talented postdoc of mine, Giovanna Di Stefano. So what are the key cellular drivers of age-associated cellular decline? We've already talked about some of these, but I'm going to go into somewhat more detail. I've mentioned the decline in NAD that is directly associated with the reduced cellular energy production. The decline in mitophagy we're going to talk about, and overall mitochondria health that relates to a reduced efficiency in the fueling of the muscle cell, providing energy to the muscle cell for proper muscle function. And then lastly, oxidative stress, this decline in glutathione. That's our body's natural antioxidant defense against oxidative stress. If you run out of your oxidative antioxidant capacity, in this case glutathione, obviously this is going to result in a greater overall increase in your oxidative stress. So oxidative stress is really the balance of production of the oxidative stress molecules, the free radicals, and the ability to deal with these oxidant free radicals through your body's natural antioxidant defense system that normally declines with age. So overall, these cellular mechanisms that impact mitochondria health associated with age-associated cellular decline, many of these center around mitochondria, which again are the energy powerhouses of the cell. Again, some key functions of their mitochondria: they can efficiently convert both fat and carbohydrates to energy. And then importantly, this NAD molecule plays an important role in producing energy, or ATP. Mitochondria production also generates free radicals. So it almost seems like a paradox, right? So you have these mitochondria that produce energy, but as a byproduct of the energy production, they produce free radicals, which in excess can be harmful to the cell. So that's why I mentioned this glutathione as the body's natural antioxidant defense mechanism against these excessive free radicals created in the mitochondria. And then lastly, these damaged mitochondria are cleared through a process called mitophagy, which is essentially the quality control degradation of the malfunctioning mitochondria. First of all, NAD, I'm not going to go into a lot of detail on this because Dr. Cheney after me will talk to you in a great more detail about NAD biology and the potential for NAD boosting as a potential anti-aging target. But first of all, I'll set it up to say that NAD is crucial for many cellular functions, including mitochondria functions, cellular energy and repair. NAD levels and mitochondria function have been shown to decrease with age or at the onset of many age-associated diseases. And this is a schematic here showing that NAD declines up to 50% between the ages of 40 and 60. So the therapeutic potential of boosting NAD in aging and age-related diseases, again, a whole host of ultimate outcomes that are potentially from a reduction in NAD with aging, DNA damage again, altered cellular communication, cellular senescence, mitochondrial dysfunction, et cetera, et cetera. So what are the factors that may contribute to either the loss or the potential preservation or increase in NAD? Fasting and healthy diet, exercise, I mentioned these NAD precursors, potentially nutritional interventions to boost NAD through these NAD precursors, or potentially drugs or other nutritional interventions to inhibit these NAD degrading or consuming enzymes. So ultimately, we want to maintain healthy levels of NAD. So I'm going to drill down a little bit more on this concept of mitophagy just for a minute here. But these are newer concepts indicating that this mitophagy, or getting rid of bad mitochondria, is a key to mitochondria and cellular health. So essentially, healthy mitochondria can lose function over time, whether this is a product of excess oxidative stress, environmental exposures, lack of exercise, etc. But ultimately, this loss of mitochondria function, you would want to target these mitochondria to basically to leave the cell. But if the cell is deficient in the ability to rid of these damaged mitochondria, this can be a problem because essentially you don't want a lot of damaged cell trash or cell waste floating around. You want a good mechanism to get rid of this. And this mitophagy, this mechanism of getting rid of bad mitochondria, has been shown to be defective in aging. And it's been shown to relate to impaired muscle function as well as predisposed to neurodegeneration. So potentially linked to both cognitive dysfunction and physical dysfunction in older age. Now, mitochondrial dynamics. So the mitophagy, the degradation of mitochondria, is thought to be linked to the dynamic aspects of mitochondria, these how mitochondria move around in the cell, how they divide, how they fuse back together. So there's a process called fusion, increase in mitochondria mass. There are proteins targeted towards this process. Fission, increase in mitochondria number, ultimately that relate to making better new mitochondria. In some of our data, in our muscle biopsy data, we've shown that compared to young people, middle-aged and older people have lower amount of proteins associated with these dynamic components of mitochondria biology, dynamic fission and fusion. But what's important is that when we separate out the young subjects into active and sedentary, you can clearly see what's driving this association between young, middle-aged, and old. It's not the age per se, but rather the level of physical activity in the older people. So physical activity is associated with a better mitochondria health, getting rid of bad mitochondria, and making new mitochondria. It doesn't seem to be associated, at least in our hands, with aging per se. Now, I've talked to you about exercise and the benefits of exercise on mitochondria, but I also want to point out that two important points here. One is that not everybody can or wants to exercise. We know that. Whether you're immobilized or hospitalized, etc. So it's not practical for everybody to exercise. Second of all, even if you are exercising, there's likely a very important nutritional component that is needed to go hand in hand to support the increased activity, to support the increased metabolism that comes with increased exercise. And this really brings me to the very last part of the talk, which is really targeted precision medicine type of nutritional interventions to affect mitochondria. Urolithin A, UA, is a novel ingredient that activates mitophagy. It's a metabolite produced by gut bacteria following the consumption of certain foods. Importantly, the urolithin A synthesis declines with age, and only about 30 to 40% of the people have the gut microbiota for efficient transformation of this. The preclinical or the rodent or the mouse data are very clear in showing that if you put these mice on a supplement of your UA, it increases both grip strength and aerobic or endurance in these mice. So what are the human data? Well, there have been very few human studies on this, but one of the important human studies has shown that if you put humans on a UA supplement, either 500 milligrams or 1,000 milligrams, it increases the gene expression associated with autophagy, mitophagy, mitochondria function and biogenesis, and importantly, fatty acid oxidation. So the take home from this is that this human translational study shows that when you put people on this UA supplement, it can have a very powerful effect to increase the machinery to oxidize fat better and to increase your overall mitochondria function. So lastly, oxidative stress and aging. It's a complicated thing because it's a balance, as I mentioned before, of oxidative stress and the antioxidant defense system. If it's imbalance, if you have an excess of oxidative stress, this will lead to oxidative damage. So you have to have the right balance of antioxidant defenses to combat the excess free radical production as we get older. And as I mentioned, if you exercise, you're actually going to produce more of the free radicals, all the more important reason to have a strong antioxidant defense system. And we think that glutathione is a key to the cellular protection. Glutathione synthesis and concentration decreases with age. It's correlated with the increase in oxidative stress markers. You can see in this clinical study comparing young to elderly, the elderly had a much lower glutathione content compared to the younger subjects, so their antioxidant defense system is much lower. So what are the nutritional strategies here that we can mitigate the naturally occurring decrease in glutathione? Amino acid precursors, glutamine, glycine, cysteine, for example, can increase glutathione. micronutrients potentially, certain vitamins, minerals. And then there's certain dietary patterns associated with increasing glutathione levels, the Mediterranean diet, the DASH diet, for example. And certainly exercise can increase glutathione levels, at least in muscle and some other cells. So I'd like to conclude by saying that aging is only loosely associated with the decline in mitochondrial energetics. Obesity and physical inactivity are much more powerful drivers of the decrease in mitochondria function in aging humans. Exercise has a much more profound effect on mitochondrial capacity and muscle compared to weight loss by calorie restriction. And then lastly, promising new dietary interventions are now available to improve human health and aging by enhancing mitochondrial energetics. So I'll be looking forward to the question and answer period at the end. And at this time, I'd like to hand it over to Dr. Cheney.
Well, I would like to thank the Nestle Health for the invitation to talk at the Aspen. I'm going to talk today about the role of mitochondrial dysfunction in disease and health, and also opportunities to approach mitochondrial dysfunction with nutritional manipulations. So first, just some disclaimers. I have a couple of patents and licenses that I need to make you aware of. The objective of this talk is to review the primary and secondary mitochondrial dysfunction and diseases, and then I'll delve into some of the specific roles of NAD, nicotinamide adenine dinucleotide, in mitochondrial function and cellular functions, and then discuss how we can manipulate NAD levels through providing derivative of vitamin B3 to try to optimize mitochondrial function in some disease states, and also to maintain health span. There is a book that I like quite a bit that is authored by Nick Lane at the UK. He has been an author that has been writing a lot about mitochondria. And one important thing is that when we learn in biochemistry, mitochondria as the powerhouse to generate ATP, and that's pretty much all we learn in biochemistry, but mitochondria is way more than that. Mitochondria is involved in apoptosis, for example. It comes from your mom, so a lot of the mitochondrial diseases are derived from the mother. There is also roles in signal transduction and in several disease states, and also in aging. Some of the functions of the mitochondria have been elucidated through the years and have actually led to multiple awards, like the Nobel Prize in 1997 for the discovery of the signal transduction that leads to the synthesis of ATP. We can't forget also Mitchell's theory of the electron gradient that's very important for the mitochondria. Now, what the mitochondria does is that it uses the energy generated from the substrates such as glucose, lipids, amino acids, and then transfer that to different oxyreduction acceptors that are then important for the utilization through the complexes in the mitochondria to generate then a proton gradient, and eventually the proton gradient is used through the F1, F0 ATPase to make ATP. But mitochondria is way more than that. So mitochondria is not only this powerhouse, but it's also, in terms of its biology, it's very important to understand that mitochondria has a life itself. So mitochondria can actually be generated through mitochondrial biogenesis, and then mitochondria can also be modified in terms of its function and also shape. and then mitochondria has to be disposed when it's dysfunctional. So all those phenomena are very important for mitochondrial function, and they can be impaired in different disease states. So, for example, on the right side, you can see some of the proteins involved here on the mitophagy. Mitophagy is actually impaired in Parkinson's disease, for example, where Parkin and pink are part of the disease state. One of the things that we have been studying, and many others have done studies on this, is that mitochondrial function and mitochondrial dynamics appears to be impaired in aging, for example. So mitochondria that is dysfunctional accumulates in aging tissue, and this appears to be related to a defect on this mitophagy pathway. There are actually new approaches now to mitophagy. I'm not going to talk a lot about it, but there is a derivative of the fruit and seed, the pomegranate, called urolithin A, that appears to be able to regulate mitophagy. So this is potentially something that one can use, but more studies are necessary to understand the role of urolithin A in health and disease. Another thing that's very important about the mitochondria is to know where is the mitochondria. So because the very energy demanding tissues need to generate ATP for its function, it is expected that heart, kidney, skeletal muscle, and the brain have most of the mitochondria. So in terms of mitochondrial diseases, both primary and secondary, a lot of the symptoms that you're going to see and signs are related with some of those tissues. It's also important to know that mitochondria is very, very important for the immune system. So here's just a description of some of the primary and secondary mitochondrial diseases. Most of the primary or genetic diseases of the mitochondria, I think is what most people know about. MELAS is a great example, and there are other types of mitochondrial diseases. Those are usually, as I said, genetic. The genes to actually make a mitochondria are not only in the mitochondria, but also on the nuclear DNA. And in addition to that, there are deletions in some of the mitochondrial DNA that can cause mitochondrial disease. But probably more prevalent is the fact that mitochondrial dysfunction is actually present in multiple secondary states. And as I said, in Parkinson's disease, there is impairment in the mitochondrial disposal mechanism, but in other diseases, mitochondrial dysfunction is also present. The primary mitochondrial diseases, as I said, they are more rare, they are genetic in its cases, and they usually happen because of mutations that happen either on the nuclear DNA or mitochondrial DNA. It is actually important that mitochondrial dysfunction happens every day. Viral infections like influenza, like the new coronavirus, dengue virus, they all can cause some degree of mitochondrial dysfunction. Your diet can cause mitochondrial dysfunction or ameliorate mitochondrial function. Even prolonged inactivity and immobilization in patients in the ICU or in patients after major surgery and fractures can lead to loss of mitochondria and mitochondrial dysfunction. There are even reports of physiological stress to be causative of mitochondrial dysfunction. Another thing that we see very often in the clinic is the use of antibiotics, and it is actually quite interesting that both the bacteria and the mitochondria appears to come from a common ancestor, And because of that, a lot of the antibiotics that work in the mitochondria on the bacteria can also affect your mitochondria and cause some degree of mitochondrial dysfunction. And there is some evidence in the literature that some of this antibiotic-induced mitochondrial dysfunction can lead to evidence of metabolic syndrome. So, as I said, NAD metabolism is very important for the mitochondria, and one of the things that it does is that it's used by one of the mitochondrial complexes to generate a proton gradient that will then generate ATP. So NAD is part of this complex 1, but NAD is important for many, many, many oxy reductions in the body. And in addition to that, NAD is also a signaling molecule for many, many reactions that happen in the nuclei, in the cytoplasm, in the mitochondria, and even outside of the cell. There is now growing evidence that changes in NAD can modulate all the way from mitochondrial function to epigenetics, to signal transduction, and circadian cycle, inflammation, so on and so forth. So NAD is very, very important for our biology. NAD can be actually synthesized or degraded through multiple pathways. And that also is important when we think about ways to actually replace NAD or cause NAD boosting. So as is shown in this picture over here, NAD is also important for multiple disease states. So NAD levels appear to decline in several preclinical models of human disease, and including aging and several age-related diseases. In the human, it's actually evident that NAD decline is important for not only the well-known disease pellagra, but also for other diseases. So in a couple of years ago, our lab and other labs have actually developed a concept of NAD boosting therapy. And through that, either through inhibition of its degradation or activation of its synthesis, or by providing vitamin B3 derivatives, we have shown, and many others have shown, that in preclinical animal models, you can increase NAD and have some salutary effects in different preclinical models of disease. Pellagra is actually one of the biggest examples of the use of vitamin B3. So pellagra was endemic in the 1800s and 1900s, especially in the Midwest, where a diet low in vitamin B3 caused this dermatitis, diarrhea, and dementia, and eventually death. Adding vitamin B3 to the diet pretty much made pellagra non-existent at this point. Also, it's very interesting that there are studies that actually show that long-term administration of vitamin B3 can even increase longevity in patients after an MI. So this is an old study in the 70s showing that as a secondary prevention, niacin, one of the vitamin B3 derivatives, could also increase all causes of mortality and increasing longevity. Very interestingly, in this study, they follow up the patients even after the niacin was stopped, and niacin was able to continue to have an effect. There's a recent study showing that actually in acute kidney injury, giving high doses of vitamin B3 like nicotinamide can decrease the incidence of perioperative kidney injury. We are actually doing a study here in Mayo right now that we're enrolling patients undergoing major vascular surgery to look to see if this is actually applicable to a large number of patients on a double-blind study. The study published in Harvard that I'm referring to was a smaller study and needs validation at this point. So it's not yet recommended, but it's something that we will know in the near future. In mitochondrial myopathy, for example, there is actually also a small study that has shown that giving niacin to patients with mitochondrial myopathy. You can have improvements also in the biochemistry in terms of NAD levels, in terms of its molecular markers of the disease. There was also improvement, but more important, there was improvement in some of the exercise performance with muscle function in the abdominal wall increasing several fold. This study, as I said, is small and needs validation, but indicates that perhaps vitamin B3 supplementation would be helpful for this mitochondrial disease population. As I said, in aging, we have actually shown that mitochondrial dysfunction and NAD levels decline over time, and this appears to be related to degradation of NAD. So NAD, as I said, can be approached through multiple pathways. And you can have NAD synthesized through a de novo pathway through amino acids like tryptophan, or through the salvage pathway, or a pathway that uses nicotinic acid. Here on the top, I'm actually showing some of the derivatives of NAD. So NAD can be derived from a precursor called NMN, NR, or as nicotinic acid or nicotinamide. Importantly, a lot of those precursors, when they're given orally, they're actually metabolized through the first passage and circulate as nicotinamide. And appears that NR, for example, can boost NAD both in the liver and the kidney, but it circulates like nicotinamide to the other tissues. There are obviously other ways that one can attempt to ameliorate mitochondrial function, and those are being explored in different labs, and there are both animal studies and human studies that support this. So, for example, caloric restriction can actually ameliorate the mitochondrial dysfunction that one sees during the aging process, and appears to be not because you make more of that mitochondria, but it's because you actually prevent damage and you clear the dysfunctional mitochondria. So there's a lot of future work that needs to be done in terms of mitochondrial dysfunction and nutrition interventions for this, but there is an emerging role, as I said, of boosting NAD levels through different pathways. So in conclusion, mitochondrial function is quite complex. There is a mitochondrial quality control that's important, as I said, in terms of its damage and its mitophagy. NAD is key for the function of the mitochondrial cells, and there is actually clinical evidence, preclinical evidence, and some human evidence that NAD boosting might be beneficial for some disease states. Just want to highlight one last study that was published just very recently, showing that in animal models of diastolic dysfunction or heart failure with preserved ejection fraction, giving high doses of vitamin B3 could correct this cardiac dysfunction. And it's a disease that has no treatment at this point, for example. All right, so now I'm gonna just kick off to Dr. Martindale for the Q&A of this section.
Great. Well, thank you everyone for listening in today, and I think we should have an exciting question and answer session here. We've got some very good questions already, 29 in the queue, so we'll move right along. Why don't we start off with Dr. Genie. What do you think about magnesium and its function in the mitochondria? What can we say about magnesium levels? You know, we hear lots about it in cardiac function and preventing cramps in the legs. Is it part of the mitochondria, the issue here?
Yeah, so I think it's important to know that a lot of the enzymes, especially they use nucleotides, they actually use magnesium nucleotides. So, for example, the ATPases or the synthesis of ATP in the mitochondria uses magnesium as one of its cofactors. So magnesium is key for mitochondrial function. And there are actually some decent trials showing that magnesium can decrease the incidence of post-op for example, atrial fibrillation and other arrhythmias. So it is, I think, a key component of the nutrition. Unfortunately, we only measure total magnesium and not free magnesium, what probably matters.
And what about intracellular magnesium versus extracellular magnesium? Do we see a big change there? And also in the mitochondria itself, is that level very different than it is in the cytoplasm?
So again, I think that the major difficulty there is what we can measure and how much of the free and actually bound magnesium we can actually see. So I think some technique advances are going to be necessary for us to really know exactly what are those pools. But I think that is a very important question.
Great. And so, Brett, we have several questions here about mitochondria and age, about what type of exercise, aerobic, anaerobic, what's best for mitochondrial function, you think? And then also, can you relate that to age? Should an aged person be doing different exercise than a non-aged person, like I'm the age group now?
Yeah, I think that's a great question or questions. I think part of it depends on sort of where your baseline or starting level is. I mean, we've done controlled exercise studies where we have older people in their 70s do brisk walking for 12 weeks and their mitochondria function improves by 50%. So, if you're starting out at a lower level, I would say, a moderate aerobic exercise program will reap, huge benefits. If you're younger and more physically fit and active, you probably wouldn't see increases in your mitochondria unless you do, more robust exercise. But I think the resistance exercise question is a really good one. We don't have a lot of data on the effects of resistance exercise on mitochondria, particularly in older people. We're doing a big study right now to get at that. So, and I think part of the answer is similar to what I give people when they ask me what type of exercise should I do for anything. And my response is the type of exercise that you'll do. Yeah. Whether it's walking or cycling, I think anything is better than nothing. So, you could slice it up and look at high intensity interval training. As long as you stick with it, I think practically any type of exercise is going to be beneficial to mitochondria.
I was intrigued with both of the talks, and should we try to push any of these cofactors like NAD or B3, B2, whatever? Is there any one of those you would say people should be taking, or should we just eat a good balanced diet?
Well, I think, the point I made in the talk, and probably brushed over it a little too quickly, but I think it's a good one to raise now, which is, even people who exercise, it's possible that they need some additional support for better mitochondrial health. Exercise is, can induce profound improvements in mitochondria function, as you and I talked about, Bob. But the question is, with the antioxidant capacity and the increased, oxidative, free radicals that are produced with exercise, could people benefit from, better nutrition, let's say, to support better mitochondria health, even if they're exercising a lot?
That's great. Okay, I see one here that I'll try to answer. Does EPA-DHA affect in protein synthesis? I think that the data in EPA-DHA affecting protein synthetic machinery is mainly on its effect on decreasing inflammatory changes. If we decrease intramuscular inflammation, like Putticherry's work has shown and several others now, we actually show then an enhancement and uptake by amino acids and protein synthetic machinery is better within the cell. So I think a lot of that is not only the inhibition of inflammation, but also the enhanced resolution of inflammation, which is shown by, Charlie Sirhan and the group out of Boston, now out of London, with Jess Dolly's work showing, again, enhancing resolution, allowing the synthetic and anabolic machinery of the cell and protein synthesis. So another one we had down here that I think I was just looking at, was quite fascinating, was with the supplemental, the mitochondrial health and polyphenols, or any of these others. Should we be doing those? Should we be trying to think about some of these antioxidants in addition to exercise? and mitochondria, look at the mitochondria? Can you bind these together? Either Eduardo or Brad, either one.
So I think that there are a couple of things there. So one is a lot of the polyphenols, I think one of the issues is bioavailability. So resveratrol, for example, that had been really publicized quite a bit and was, again, going to save us all from everything. It is really,
the red wine.
Yes, the red wine. So it was it's metabolized really fast through the first passage, and bioavailability is poor. So that's one of the issues with a lot of those compounds. Mm-hmm. The other one, as you said, is that blocking ROS is not necessarily a good thing all the time. And exercise, as has been shown before, to actually be effective in both mitochondrial activation and also increasing insulin sensitivity, needs some ROS production. So it's a balance. And I think we don't know the answer yet.
And Brett, anything different?
Yeah, just to reiterate, there was a very interesting study done in Germany several years ago where they gave participants undergoing an exercise program high levels of vitamin E and vitamin C, and it prevented a lot of the beneficial effects of exercise. So I think it's one of these Goldilocks phenomena, right? So too little is bad, too much is bad. You got to have just the right amount for some of these things.
Yeah, that's great. My next question was going to be, what about if we talk about too much of a vitamin or a mineral or whatever, we also know obviously the interactions are key. But what about the minimum or maximum exercise? Can you push too hard? Assuming you're getting adequate nutrients, can you push too hard on the exercise side?
There have been, a couple of sort of overtraining studies that have, I know one in particular, done several years ago. They used non-invasive NMR spectroscopy, the P31 method, Bob, that you raised during your talk, the non-invasive method to look at mitochondrial function in vivo. And they tested people, I think it was after a marathon or after maybe it was high intensity, short period of training. And it basically, it really screwed up the mitochondria. So there probably is some really high level of exercise that's detrimental. Having said that, I think you have to get to fairly high, levels of exercise for that to happen.
Yeah, and actually, just to echo that, so there was actually a paper just published last week on cell metabolism showing that athletes doing several cycles of high intensity training will eventually develop some mitochondrial dysfunction. Again, it's what Brad said. Would that be prevented by optimizing nutrients? We just don't know at this point.
Yeah. And is the mechanism? We've all heard about this mechanism where we need a little bit of oxidant stress to make the mitochondria undergo biogenesis, to make them so they become more active, more functional. Is that the only mechanism we know to push it? What's a way how do we increase our mitochondria other than just getting a little bit of stress to them and let them recognize we need training?
Well, I think the question about, what is the basic mechanism by which exercise improves mitochondrial biogenesis is still really not all that clear. We know that AMP kinase, increase in AMP levels during exercise probably has something to do with it. As Eduardo knows, calcium during muscle contraction has a role in mitochondrial biogenesis. But there, probably some many things that we don't yet know about.
And I think just also to add, so the NAD sort of boosting, we know that in cells and in animal models, through activation of the SIRT1 pathway and probably SIRT3 pathway, activates this PGC1 alpha mediated mitochondrial biogenesis. So mitochondrial biogenesis it happens in multiple steps, and there are multiple pathways. But as both you and Brett said, it seems like there is a signal to say there is a stress, we need more mitochondria.
Yeah, and I would also add that, each of us, I think, touched upon it a little bit, was, this concept of mitophagy or autophagy. So there's some evidence suggesting that if you have a defective ability to get rid of bad mitochondria, that also directly impacts your ability to make new mitochondria. So, it sort of goes hand in hand, right? If you can't get rid of the bad mitochondria, you're not able to make new ones. So let's not forget that process. And maybe there's some, nutritional interventions that can aid in this autophagy process. So that's really brand new territory.
Great. And, the couple of questions on this idea of metabolic reprogramming and bacterial versus viral illness, in the ICU setting, which I mentioned by that article, showing that in the bacterial models, giving nutrients early on was more detrimental. And if they were not able to use glucose, and giving glucose made them sicker and died of sepsis, actually. Whereas in the viral model, they could upregulate the glycolysis and had to have glucose, otherwise they failed and had high mortality. So it was very interesting to me in that what do you figure? You think this is a mitochondrial problem? Because when glycolysis is activated, when inhibit glycolysis, we're forced to use then fats or, at least ketone bodies. So do you think we should be trying to manipulate the metabolism in different illnesses? Like maybe take a look at the patient, see there's some marker that would say, this is the patient who's going to use glucose, or this is the patient who needs keto acids or fatty acids. Do you think there's any role or potential there?
I think it's a great question, and sort of to piggyback on that a little bit, Bob, I think, the other question for me is how does your systemic metabolism, whether you can shift glucose metabolism or fat metabolism, ketogenic diet, for example, we know that it has profound effects to shift from carbohydrate to fat metabolism. How does that affect the metabolism of the immune cells? Is there a direct connection there between how the immune cells utilize energy and their function, right? So I don't think we know anything about that.
Yeah, that's a good thought. And that brings up the point that one of the questions here was about to Eduardo. What about, you mentioned the mitochondria affects the immune system. Can you elaborate on that a little bit and how that works?
Yeah, so in fact, it has been shown that there are big shifts on metabolism and mitochondrial function during activation, for example, of T cells and macrophages. So a macrophage, when it goes from a non-inflammatory macrophage to a pro-inflammatory macrophage, uses more glycolysis and kind of shuts down its oxidative phosphorylation. This doesn't seem to be a dysfunction. It's actually really an adaptation, because it ends up producing other compounds from the Krebs cycle and don't actually go through the oxidative phosphorylation. One other thing that I think it's really cool to think about, the nutrition during different types of infections, is that if you think that mitochondria, and that is the hypothesis, mitochondria and bacteria are coming from a common ancestor, right? When you're trying to feed your mitochondria during a bacterial disease, you're feeding the bacteria too. So you might be losing there because you are giving the energy to the bacteria and not getting to your mitochondria, for example. One of the key things is, for example, in terms of NAD, there's actually some bacteria cannot make NAD at all. So they actually have to take full NAD from the environment. So when you actually do that, you might actually be making things worse. So
this sort of brings up an interesting part of this whole microbiome question. Everything nowadays wants to see what's the effects on the microbiome, or do the microbiome affect our mitochondria in any way? Have you seen much on that? Is it metabolomics of the microbiome can affect our mitochondrial health, whether it's a gut mitochondria or the liver mitochondria, or whether it's systemic mitochondrial effect? Anybody got any thoughts on that?
I think that the paper actually that Brad mentioned is exactly on that, the urolithin A is actually produced by the microbiome. There are people that don't have the bacteria that can make urolithin A. And then that urolithin A that's produced by the bacteria goes and changes your mitochondrial dynamics. And this, I think, goes back to this concept of hormesis, right? That there are signals within ecosystems where one organism tells a different organism what's going on the ecosystem. And I think that that's what this connection between biome and our cells is.
Great. Well, we got about 3 or 4 minutes left. So what's your take-home message for people today? First, you'll go with Eduardo. You have a take-home message? How do we keep our mitochondria healthy?
I think as you was mentioned, I think exercise, proper diet. Do we know exactly what the best diet is? We don't, but Mediterranean diet seems to be ideal. Moving, even if it's not exercise. So for example, there's a lot of things showing that there's this no-exercise thermogenesis that is beneficial to you. And, I think that we need to learn a little more about optimizing other nutritional supplements. There's a lot of preclinical data and even human data that supports that some of those factors, like, let's say, vitamin B3, can actually modify things in beneficial ways, for example.
Brett, Nate,
and I would just add quickly, in addition to that, first of all, clearly exercise is probably the most obvious way to increase mitochondrial health. But I would also, again, highlight the potential importance of non-exercise therapies, either as an adjunct with exercise or maybe even, independent of exercise for those people who can't exercise. I mean, for example, we've done bed rest studies, and the most profound effect of disuse with bed rest in people is on mitochondria. So, identifying nutritional interventions to support that. And I think the question that came through in the chat about, chemotherapy. I mean, chemotherapy is a poison to mitochondria. We're doing a study right now to look at the effects of chemotherapy in cancer patients on mitochondria. So really try to identify, non-exercise therapies that may mitigate or prevent that will be key, I think.
Yeah, I believe in my sort of review of the literature and my limited knowledge of biochemistry nowadays, current biochemistry anyway, it looks like this is a wide open field. And I think it's Eduardo and Brett, you guys are certainly on the cutting edge here, and it looks like an exciting place to be at the really the precipice of really seeing some changes and some maybe use clinical tools or chemotherapy tools or aging tools are going to be in the mitochondria. That may be the fountain of youth if we can keep a healthy mitochondria, And so I think we've got a lot to learn about this, and I guess we're pretty much out of time. It's I guess my time, 2:45 or 5:45 for you guys. We're certainly glad appreciate you joining us. And Eduardo and Brett, I'm fascinated by your guys' work, and I've got a lot of reading to do to catch up here. Thank you very much for helping us out today with this conference.
All right, thank you, guys.